Hi, pt.1 students. So today we're going to talk about DNA, RNA, and the history of it. So first, some basics. DNA, let me switch to a new layer. DNA, which stands for deoxyribonucleic acid, and RNA, which stands for ribonucleic acid, are both nucleic acids, which is one of the four main macromolecules. The main macromolecules of biology are nucleic acids, proteins, lipids, and carbohydrates, and they are made of the monomer nucleotides. And remember, nucleotides are monomers of nucleic acids. Monomers are the smaller building blocks that make up polymers. So DNA has the simple sugar deoxyribose, while RNA has the simple sugar ribose. And the difference between deoxyribose and ribose is that deoxyribose is just missing one oxygen. It has one less oxygen compared to ribose. And then DNA has four nitrogenous bases: adenine, thymine, cytosine, and guanine. And adenine always bonds to thymine, and cytosine always bonds to guanine. And remember, the apple goes in the tree and the car goes in the garage. And RNA is the same thing, but thymine is replaced with uracil. So in terms of their shapes, DNA is a double helix. Let me see if I can draw this out. You guys have probably seen this before. It just looks like this. And then it has like these rungs. Let's just say that's A and that's T, and then they're hydrogen bonded together. And there's just like a bunch of rungs on DNA. So that's like the shape of it. RNA has three different shapes. Messenger RNA is linear. It kind of looks like this. Transfer RNA is like a clover leaf shape. And then ribosomal RNA is like a globular shape. It kind of just looks like this. So messenger RNA, its job is to take instructions to make a protein from the nucleus to the ribosome. So basically, it floats out through one of those nuclear pores, and then it travels to the ribosome. It travels to the cytoplasm, and then it attaches itself to a ribosome. The ribosome reads it to make proteins, but we're going to talk more about that later. Transfer RNA brings amino acids to the ribosomes. Amino acids are usually just floating around the cytoplasm, and transfer RNA brings the amino acids to the ribosomes to form a protein. And then ribosomal RNA makes up the small subunit of ribosomes. So ribosomes, here's like a very rough drawing of it. It kind of looks like a hamburger bun. There's a small subunit, which is this part, which is made of rRNA, ribosomal RNA, and then there's a large subunit, which is this part, which is made of proteins. Okay. And now on some history of it. Surprisingly, on the AP Bio test, there are a few history questions. It's just basic level history. You just need to know some famous scientists like Charles Darwin, Mendel, and Watson and Crick. So it all starts with the Nobel Prize. Basically, there was a guy named Nobel. He invented dynamite, and then he felt really bad because people were using it to harm each other, so he wanted to do something good for humanity. So he came up with the idea of the Nobel Prize. And in order to get a Nobel Prize, you must be alive, you must win $1 million, you must be alive, you must help humanity, and you must make a big contribution in your field. And if you win the Nobel Prize, you get $1 million, and you get a lot of prestige in the scientific world. And some examples of fields that you can win a Nobel Prize in are medicine, biochemistry, or chemistry. I don't think there's any biology field. There's biochem, but there's no biology field. So there's a Nobel Prize on the line to find the shape of DNA. And so scientists around the world were all racing to find the shape of DNA. And a woman named Rosalind Franklin used something called X-ray crystallography, which is just a fancy word to say a fancy type of imaging process to show that DNA was a double helix. Basically, she took a picture from the top view. So DNA is a double helix, so from the top it kind of looks like an X, because that's like the double helix part. So she took a picture of the double helix. You can Google an image if you want to see her pictures. And a man named Wilkinson, working in the same lab as her, shared her work to a rival group, Watson and Crick. They were her rival groups, and sadly, Franklin passed away due to cancer, and dead people can't win a Nobel Prize, so she wasn't able to get a Nobel Prize for her work. But basically, Watson and Crick stole her thunder, and Watson and Crick also found out some other things. They applied Chargaff's rule, and Chargaff's rule says there's equal amounts of adenine and thymine and equal amounts of cytosine and guanine. A way that it can be formulated on the AP test, is that you have, let's say you have 20% adenine. How much percent of thymine, cytosine, and guanine do you have? Okay, so Chargaff's rule says there's equal amounts of adenine and thymine. So that if you have 20% adenine, you have 20% thymine, which means you have 60% left over shared between cytosine and guanine. And there's equal amounts of cytosine and guanine, so that means you have 30% cytosine and 30% guanine. But what Chargaff didn't realize was that the bases were actually bonding, so Watson and Crick figured that out. Watson and Crick also figured out DNA replication, which is when you're taking one molecule of DNA and using it to make 2 molecules of DNA, and we can talk about that in our next video. And Watson and Crick also proposed a semi-conservative model of DNA replication. So basically, new DNA molecules are half made of new nucleotides and half made of old nucleotides. So DNA is a double helix. And then one strand is going to be made of new nucleotides and one strand is going to be made of old nucleotides.